Okay, everyone, thanks for coming. I'm pleased to welcome Kit Yates to give us a lecture today. He's a very talented mathematician working in Bath University at the minute, where he's got a lectureship. He works in the area of mathematical biology, and so he's going to talk to us today about his research in multiscale models, in particular in cell migration and also in patterning in development. Welcome, Kit.
Great, thanks. So thank you very much for the invitation to come and speak to you guys today. I'm going to talk to you about some multi-scale methodologies for representing cell migration, but with the idea that they might be extended to more general stochastic biological processes, maybe even general reaction-diffusion type processes as we go along. So I'll start by giving you a sort of brief outline. I'm going to talk in two parts. The first part is going to be looking at an actual biological application, modelling neural crest cell migration. Talk about what neural crest cells are, why we're interested in modelling their migration, and discuss the experimental evidence that we have in order to parameterize our models. And I'll show you how a really simple, straightforward mathematical model, one of the most simple ones you could write down, can actually be quite useful for generating some biological insights and answering some biological questions which are perhaps less accessible to experiments. And then I'll show you some of the results from the model. And the first part will then act as a sort of motivation for the second part, which is going to talk about developing hybrid models in order to accelerate spatially extended stochastic simulation. So I want to scale these individual based models up. They become very computationally inefficient, so I want to take two different modelling regimes, one which is efficient but maybe not as accurate, and one which is more accurate but maybe very computationally inefficient, and hybridize them together in space. So I'll talk about different modelling paradigms, different ways we model diffusion or reaction diffusion, some algorithms that we've come up with to couple these things, and then I'll show you that at least in one of the cases that I will show you that we can actually show that it works. Then I'll talk about where we're going to go with this in the future. So for the first part, the results of this part are in this Nature Communications paper, which we published back in January. So this part is looking at modelling neural crest cell migration. So the first question that I like to address is why we should bother looking at neural crest cells in the first place. So neural crest cells are a diverse range of cells in the early embryo. They come from a region called the neural crest, which is just dorsal or just behind the neural tube, which goes on to become the spine in invertebrates. Sorry, in vertebrates, not in invertebrates. Obviously, invertebrates don't have a spine. That would be confusing. So different types of neural crest cells go on to become different types of tissue in the adult. They contribute to parts of the bone, of the gut, the eye, the heart, and importantly for my applications, the skin and the hair. If something goes wrong with these cells, their migration or their proliferation, then you have a whole family of diseases which are called neurocrestopathies. So these range from the very mild end, things like piebaldism, which is what I'm going to talk about today, through to things like Hirschsprung's disease, which is a failure of neuroblasts to colonize the appropriate domain, which means your gut doesn't get innervated properly, so you get chronic obstructions of the gut. And even further extreme things like neurofibromatosis, which is sort of predisposition to cancer. Okay, so they're an important set of cells, and if something goes wrong in the migration, then you get a whole range of different diseases. In particular, I'm interested in a subtype of neural crest cells which are called melanocytes. So these are the cells which go on to produce pigmentation of hair and skin. So these melanocytes, they migrate from the back of the embryo around to the front, and we're particularly interested in the region between the limbs, so it's the trunk region. And they start off with a small number of cells near the back in this region here in red, called the migration staging area. And over a 5-day period, these cells proliferate and they migrate, and they colonize the whole of the skin over a 5-day period. So from embryonic day 10.5 to embryonic day 15.5. So here we're seeing a cross-section of this. So this is the outside of the mouse embryo. This is a picture of a mouse embryo. And so the cells migrate around the somite, around the somites rather, and through the developing dermis and epidermis, through the developing skin, if you like. So that's the sort of some of the biological background. Why am I interested in melanocytes in particular? Well, they have this pathology if something goes wrong called piebaldism. Piebaldism is in some senses a very mild disease. You can live perfectly normally with it. In fact, many humans have piebaldism. I can't confirm this because they haven't had a genetic test, but David and Ed Miliband both have a white forelock of hair, which is quite characteristic of piebaldism, so maybe they have piebaldism. It has a very clear phenotype. It's very easy to see when an animal has piebaldism, and therefore if you make a change to something in the embryo, it's very easy to see how you've affected the phenotype. You don't have to do anything invasive to understand whether or not this animal is showing the disease phenotype. It's a pigmentation pattern defect, so you can just look at the animal and see what happens. It's non-fatal, so you can tweak things and see what happens to the adult mouse as it grows. And the idea is that although this disease is not particularly serious, trying to understand how melanocytes migrate correctly is a sort of model for the other types of neural crest cell migratory defects, which I mentioned, which are potentially more serious. So if we can understand something in this system, we can maybe transfer our knowledge into the other types of neural crest cells, which are potentially more important. So these are as an example of the imaging that we can get from my experimental collaborator, a guy called Richard Mort, who just moved to the University of Lancaster. So these cells are stained, or not stained, they're expressing yellow fluorescent protein, and he can take time-lapse images of these cells. He does what I refer to, perhaps incorrectly, as ex vivo imaging. So it's not in vivo, which would be in the mouse itself. It's not in vitro, which would be taking some cells and plating them and watching them grow. What he does is to take some skin from the mouse, he resects the skin, and he maintains it in physiological conditions under a microscope. So somehow it's still in its biological environment, but it's not actually in the mouse itself. So it's sort of referred to as ex vivo imaging. So we can get really good data from these sorts of images, and I'll talk about that in a second. So there's a gene which causes piebaldism, and the name of the gene is KIT, which is nice because I can pretend that I discovered this gene, which I should point out is absolutely not the case, although I would like it to be. I'd like to have a gene named after me, but I'm apparently too late for that. So KIT is associated with the proliferation and survival of melanoblasts in the developing epidermis. There are a lot of different mutations for this particular gene which cause these white spotting phenotypes. So here you can see it in the mouse. He has a white belly and a white forelock, and same in the human baby here. It's a white belly spot and a white forehead spot. And that's typical of a piebald phenotype of a KIT mutation. It's caused by the fact that these melanocytes fail to fully colonize this growing embryo. So you just don't have pigment cells at the front, which is why you have these unpigmented or white regions. And interestingly, even in wild-type mice, that is mice that don't have this mutation, sometimes spontaneously you see these white belly spots appearing. So it hints that this process of cell migration, the cells migrating around from the back to the front to cover this developing embryo, is stochastic. There's a random element. Sometimes, even when all the parameters are correct in the animal, sometimes you don't get colonization. So there's a stochastic effect in here. So it hints that we're going to need a stochastic model to try and represent this behavior. So at the start of this project, our collaborator had done a lot of these movies, and he'd sort of characterized them, and he said they had these hypotheses. And they were fairly straightforward, but he wanted the mathematical model to test them. So he said, could colonization, so the cells migrating all around the embryo and colonizing, uniformly patterning the skin of this embryo, could it be explained by purely random migration, proliferation of cells, and domain growth of this embryo? So in particular, he said, I don't think I'm seeing any complex cell-cell interactions. I'm not seeing adhesion or repulsion. I'm not seeing response to signaling gradients. The only way these cells interact is through excluding volume. They effectively bump into each other and don't allow each other to occupy the same space. That's all that he had in them, all that he could see. And so he thought, yeah, maybe this could happen, but I need a mathematical model to test this out. The other thing that he noted, and this is was controversial at the time, was that when he did experiments with the cells which had the KIT mutation, he didn't see them moving more slowly. If anything, he saw them moving more quickly. What he saw was that they were proliferating more slowly, so not producing daughter cells as fast. And that's sort of interesting because naively you would expect if the cells, if you don't get to colonize this belly region at the front, which is furthest away from where the cells start off, naively you think maybe the cells just aren't migrating fast enough to get round to the front and colonize the embryo. But actually his hypothesis is that the cells just aren't proliferating enough, so there aren't enough cells overall in the embryo to colonize the embryo. So these are the sorts of things we wanted to test with a mathematical model. So he was really thorough and came up with a lot of decent experimental evidence. So here's a sort of time-lapse image of the image that I was showing you before, and you can quite nicely track these cells. They don't bump into each other or occlude each other too often, so you can get good long tracks. So here, the red curve here is an example of a track. They can be much longer than that, but it's just a few frames of this movie. And he's also been able to characterize, so you see these blue lines on top of the yellow. I don't know how well that's coming out, but he's characterizing something called the ferret's angle, the Ferret angle, depending on where you come from. And this tells you, the long axis of the cell effectively tells you the direction in which the cell is migrating. So this can tell us something about whether there is a directed migration of these cells or not. And so this is the sort of data that he can get out of these videos. So there's something called a pair correlation function. This tells you how closely, it tells you something about the distribution of these cells. So you effectively count up the distances between pairs of cells, and you compare that to the expected frequency of cells at those distances if you just put point particles down uniformly at random in a surface. So if your cells are uniformly spread across this surface, you'd expect to see a pair correlation function of 1. You just divide the number of pairwise distances by the one that you can generate by just putting these points down uniformly in the region. And so we basically see that for most distances the cells are uniformly distributed, apart from when you get to very small pairwise distances. And actually you see fewer pairs than you would expect if these cells were points and they were uniformly distributed across this region. And the reason for that is that these cells do actually exclude volume. So the point where this starts to dip down is actually around 30 to 40 microns, which corresponds roughly to the diameter of a cell. So you'd expect to see fewer pairs at those shorter distances. So that's something interesting. We can characterize that, yes, generally these cells are uniformly distributed in this tissue, but occasionally they don't tend to get too close to each other because that's the radius of the cell is volume excluding. You can take the tracks and you can plot them all with this common origin, and you can do various isotropy metrics on these, so you can see that these cells don't tend to be migrating in one direction over another one. There's no anisotropy in this system. Okay, this is just me plotting the tracks and arguing to you that it looks like there's no anisotropy, but you can actually do proper metrics to test that that's the case. And then you can do things like measuring the mean squared displacement of these cells. So if these cells are truly diffusing, you expect the mean squared displacement to be linear, and you expect it to be through the origin. And that's pretty much what we see with our data. So here, red line is just the best fit, and black is the actual experimental data. So we're fairly confident these cells are not moving in a directed manner, and indeed that they are diffusing. So that's quite fortunate from a mathematical point of view, because there are quite a lot of well-characterized models of diffusion, so we can go ahead and model these cells as if they're diffusing. We also have these relationships from the data where we look at how the diffusion coefficient varies with cell density, and how the cell cycle time varies with cell density, the cell division time. So the black dots here are the experimental data. So you can see that as the cell density increases, the diffusion coefficient tends to decrease with these cells. And similarly, as the cell density increases, the cell cycle time increases. So proliferation slows down as the cell density increases. So these red curves are not just arbitrary best fits that I've put through them. In fact, they definitely aren't best fits. In the model that I'll tell you about in a second, we have two parameters. One controls movement, and one controls proliferation, how fast these cells divide. And actually all we've done is to choose our diffusion coefficient to minimize some of the squares of the residuals. So we've done a least squares fit for this one parameter. But the fact that we have this same density-dependent relationship for the diffusion coefficient and for the proliferation time is an emergent property of the model. This is not something you have to put into the model. You specify the model, and this actually comes out of it. So all we've done is to change the parameter, which effectively just shifts this line up or down, and the same for this line. The parameter just changes this up or down, but the actual relationship itself, where the cell cycle time increases as the density increases, is an emergent property of the model. So it's quite nice that we haven't done too much fancy parameterization of this model to get it to look like the experimental data. A little bit more straightforwardly, we also have data on how the domain grows. So this is how the domain width increases with time. This is how the domain length increases with time. So the black dots, again, are experimental replicates. The red crosses are the mean of those experimental replicates. And then the red line is just a best fit through those. And so we can tell our model to grow in the same way. So we have two more parameters, growth of the width and growth of the length of the domain. So that's the experimental data. So we can basically characterize the model quite nicely from this experimental data without any free parameters left over to tweak. The other thing that our collaborator was interested in investigating was these stripy patterns that you occasionally see on chimeric mice. So chimeric mice, for people who are not biologists, this is my understanding of it anyway, is where you take two very early stage embryos and you mash them together. That's not a technical term, but I'm led to understand that is what you do. So you take an embryo which produces black pigment cells, and you take an embryo which doesn't produce black pigment cells. It still produces pigment cells, but they just don't have any actual pigment. They don't produce pigment themselves, so you get white areas of skin where those pigment cells are, and you put the embryos together in an early stage, and the idea is that half of the pigment progenitor cells will be producing black pigment and half won't be producing pigment. And you look to see what patterns form. And so this area here, from back round to the front, is a sort of black stripe. This is a white stripe, a black stripe, and a white stripe. And so you can see these bands of colour extending from the back to the front. And people for a long time, these experiments were done back in the 60s, and people for a long time took this as evidence of directed migration of these cells. So they must be migrating from back to front and dropping their progeny off as they go to form this stripe. And as I've mentioned, our collaborator doesn't think there's any directed migration in this. So somehow there's a contradiction in here, which we wanted to try and resolve with our model. There's also more recently been evidence where you can track the progeny of a single cell in the embryo. So you inject a chemical at a particular time, it turns on a marker in the cell, and you can watch its progeny and where they go. And this seems to support my collaborator's hypothesis that actually there isn't directed migration. These colonies of cells that initiate from a single initiator progenitor cell are actually quite diffuse and spread out across the width of this embryo, across the region of the axial region from between the limbs. So it seems to contradict these quite tight stripes that we were seeing in those chimeric mice. So we wanted to try and investigate this and see if we could replicate this in the model. So that's a lot of biology and a lot of data, but I think it's important to try and show what we've done. But now I'm going to tell you about the model itself. So it's really simple. It's an on-lattice volume exclusion model. So we take the region we're interested in, from the back to the front between the limbs. We unwrap that into a rectangle and a plane, and then we put a lattice on top of that, and we put cells in the sites of this lattice, and we say at most one cell can sit in each site of this lattice. And then I have a movement rule. So if a cell gets selected to move, so red is a cell here, black is empty space. If a cell gets selected to move, which happens with a particular rate, then it can move to one of its 4 neighbouring sites, provided those sites are unoccupied. If the site's occupied, then that movement event gets aborted. Okay, so that's the movement rule. The proliferation rule is really similar. Again, blue now is a cell, and black is empty sites. If a cell gets selected to proliferate, which again happens with a certain rate, it's one of the parameters that we specify in the model, then you can place a daughter cell in a neighbouring site. And if that site is occupied again, you abort that proliferation event, so you just don't do it, and you move on to the next event in series. So you simulate this as a continuous time Markov chain. So that's proliferation and movement. And then I said that there was also domain growth. So this is slightly more complicated. We want to try and make sure this domain grows as a rectangle because it's mathematically convenient. It's a pain in the ass if it doesn't. So what I want to do is to grow, if I want to grow in the horizontal direction, I want to, for each row that I have, I want to insert a single site into each row to make sure that I get a whole new column at the end. So what I do is, for each row, I choose uniformly amongst the columns in that row, so that's what these green sites are supposed to indicate. For each row I choose independently, uniformly, a column, and then I insert a new site into the space where I've chosen to grow, and I push everything that was to the right of that, including the original site, along to the right. So here This is supposed to indicate that I've got a cell here, and I also have chosen this site to grow. And you'll see in the new configuration that this cell has been pushed across to the right, and all the other boxes have been pushed across to the right to make space for this new empty site. Similarly, at the bottom here, I insert a new empty site here, and I push everything along to the right. If I want to do growth in the vertical direction, then I do exactly the same thing, only I choose a row uniformly at random for each column. Okay. Any questions about that? Yeah, go for it.
The new sites that you insert are always empty?
The new sites insert are always empty, yeah. Exactly, yeah. So I always shift out the cell to the right when I insert a new site. Yeah, I should also be careful because this is a cellular automaton, but the cells are actually sites or voxels, not cells as in biological cells. So that if I do mix that up, feel free to call me out on that. Okay, so I'll just show you briefly some of the results from the paper. So we first tried to simulate some of these clonal patterns from the more recent experiments. So we just tag a single cell and we watch where its progeny go. And you can see just qualitatively that it looks like we're not necessarily getting directed migration. These cells are spreading out across the axis. And if you plot this back onto an actual embryo, then this is what these colonies look like. So it doesn't really look like we're getting stripe formation on this embryo. We're getting quite diffuse clonal patterns being plotted. So that's sort of qualitative and not a bit hand-wavy. Interestingly, we can actually replicate the stripes. So it may be a bit difficult to see from the back there, but I've taken the initial cells. There are 21 of them initially on average. I've taken them and I've coloured half of them grey and half of them black, and I've made sure I colour all their progeny the same colour as the parent cell. And then I've colonised this domain, and I've looked to see what patterns occur. And occasionally I see a stripe. So I've got down here, I've got sort of mostly grey cells, and up here I've got mostly black cells. And we tried, I mean, this is a bit qualitative, so we tried to quantify this a little bit more. We came up with this thing of a clonal signal. So if you give all the black cells a value of +1 and all the grey cells a value of -1, then you sum along each one of these rows and divide by the number of sites in a row, then you get a signal which is between +1 and -1. And you can see that as the axial direction varies, so now the x-axis on this figure corresponds to the y-axis on this figure, you can see that I go from having mostly grey cells up to having mostly black cells. So there is some sort of sigmoidal shift in the value of this clonal signal, and you can do fast Fourier transforms on this to calculate the dominant modes of this pattern. But the really interesting thing is not that, I mean, I can characterize a stripe, that's sort of interesting when that happens at all, but the really interesting thing is what do these stripes correspond to? Where do they actually come from? Are they the progeny of a single initiator cell? And the answer to that is no. So if I actually colour all of these 21 cells a different colour initially, and I colour all of their progeny the same colour as the initiator cell, then I come up with a picture which looks like this. So I've got 21 different colours in this picture. And this corresponds to this stripe figure. So actually, you can see that the black stripe is made of some orange and some pink and some purple and some, I don't know what that colour is called. And then the grey stripe is made of some green and some yellow and some dark blues. So actually, the stripe formation mechanism is not directed migration. Each stripe doesn't correspond to a single initiator cell. Actually, the stripe formation mechanism is just the random coalescence of like clones which are randomly coloured one of two colours. Okay, so that's why you see the stripes occasionally in this pattern. The other important thing this uncovered was that occasionally you do see these striped patterns, but actually there's a huge reporting bias back in the 60s, and that it's not very interesting to publish a grey mouse. But people were selecting these stripy mice and they're saying, wow, look at this, it's interesting, we can draw some scientific conclusions from this. But the majority of the mice that people were seeing were not actually interesting and not stripy, they were pretty much just homogeneously coloured grey. So it's a sort of interesting thing that a mathematical model can give you, which an experiment potentially can't, because you can go back, as well as just colouring them two colours, you can colour them all different colours and actually see where each family of cells comes from. So that's the striping, and then the last question we wanted to address was this colonisation. Do I get these white belly spots appearing or not? So first thing we did was to change the diffusion coefficient in the model, and we found that we could lower the diffusion coefficient, the movement parameter, by an order of magnitude, and still get colonisation of this domain, so still get mice without this white belly spot. But as soon as we started to tamper with the cell division time to make that longer, then we found that we were starting to get these white belly spots. So what you're seeing here is 4 repeats of our simulation, and this time you're seeing dorsal to ventral to dorsal, so you're seeing all the way around this mouse embryo. So you can see 4 repeats with the wild type cell division parameter, which was a minimum cell cycle time of 8 hours. As soon as you start to increase that minimum cell cycle time to 9 hours in the model, then you start to see these white belly spots appearing. And as you further increase it to 10 and 11 hours, then you start to see these white belly spots appearing almost every time. Indeed, sometimes you even see these white back spots, which is an interesting thing that came out of the model, which I didn't really, I thought, oh no, we've got a problem here because we've got white back spots. But actually, occasionally, with these KIT mutants, you do see white back spots as well as white belly spots. So it indicates that, at least in the model, the colonization of the domain depends more on the proliferation rate than it does on the diffusion rate, and that sort of chimes with our experimental collaborators' experimental data that he had. So if you do want to know any more about this paper, then you can read it here in Nature Communications. It should be 2016 actually, but yeah, that came out in January. And so Sarah Jane mentioned that she wanted me to say a little bit about the media sort of work that I've been doing on the back of this. So although all the experiments we did were in mice, the patterning mechanism, the way that these melanocytes migrate from back to front is common to all mammals, and we know that the internet really loves a cat, so we decided to sell this story in terms of cats when we wrote the press release for this. So the Telegraph rang us up and they did a really nice chat with us. They spent like 20 minutes talking to us and wrote a nice article. The Guardian did the same. They chose to pinch it on horses, which is fine because people like horses as well, and they're quite common proponents of the piebald pattern. The Mail didn't ring us up, and they did a fairly crappy story, but it is the biggest online news magazine in the world, so we didn't complain that we got in there, and they represented things fairly faithfully. The Express seemed to think that we were going to grind up black and white cats to make medicine, and that was absolutely not what we were doing. The point was that we could use some of these mathematical models that we'd done for piebaldism to investigate other types of neural crest cell migratory defect, like I told you at the start. And then this was my favourite one, was this article from a Dutch magazine which translates as "Scientists Unravel the Mystery of the Hitler Cat," which again I should emphasise was 100% not what we were trying to do in this study. We didn't even look at cats, so I had to fight a little bit of a rearguard action when I did a couple of radio interviews with presenters saying, so why are you killing so many cats? And yeah, no cats were harmed in the making of this scientific story. Interestingly, though, if you want to scale up these models to actually say something about cats, cats are a lot bigger than mice, as are humans, as are cows, which also have interesting pigmentation patterns. People are interested in knowing where those pigmentation patterns come from because they can be commercially valuable. So when you try to scale up from Vitruvian mouse to Vitruvian man, you find that you have a problem because the mice are a lot smaller, and our computational simulations of this individual-based model are already quite expensive to run. So this is going to be motivation for the second part of the talk, which is going to be about developing hybrid models to try to accelerate these spatially extended stochastic simulation models. And there's a few references down here to the type of hybrid models that I'm going to talk about. So firstly, what do I mean by hybridization? Because different people mean different things when they talk about hybridization. So for me, I need to come up with 2 different models. One of these models needs to be coarse, maybe inaccurate, you could say, but cheap. The other model has to be more accurate but more expensive. So that's my basic setup for having a hybrid model. I need to establish something that I call an equivalence framework between these 2 models. That is, for example, I could say that if I had an individual-based model and a partial differential equation, which I thought was the cheaper, coarser equivalent, that partial differential equation maybe should describe the evolution of the mean of the population of the individual-based model. Maybe that's just an example, but that's perhaps what I should do if I want a PDE to be my coarser model. So there has to be some equivalence between them. The partial differential equation has to be doing something similar to the individual-based model. And then you need to decide where in space you're going to want to use each of these models. And then you have to come up with an algorithm to couple these 2 models together. So, for example, how do I turn PDE mass into individual particles? How do I take individual particles and make them part of my PDE? And so the example that I like to talk about or like to imagine is a travelling wave. So you imagine you've got a travelling wave of maybe cells. They're perhaps proliferating and diffusing. And behind the wave, you've got high populations of cells or particles or whatever it is. So you can get away with modelling this using a partial differential equation. Maybe you don't care about the stochasticity behind the wave. But in front of the wave, where the wave speed is being determined, you really need to get the biology or the physics right. So you need to be doing your individual-based model in that region to make sure that you're getting the wave speed correct. But behind, you don't really care so much about the details. So you can skip doing all these fine-grained interactions and just simulate a PDE, which can accelerate your stochastic simulation significantly. So that's the idea to keep in mind as we go throughout the talk. Okay, so I will talk briefly about different modelling paradigms for diffusion, because diffusion is the example that we're particularly interested in. For example, in the biological example, I said that the cells were diffusing. We'd like to build in more complex reactions to make this more general for reaction diffusion systems in general, but we haven't quite got there yet. So there are three main modelling approaches. They're often known as microscopic, mesoscopic, and macroscopic. So the microscopic models are the most fine-grained ones. So they're often called molecular-based approaches, where you model each molecule or particle as an individual. It can move around in space according to maybe Brownian motion if we're talking about diffusion. And then they can bump into neighbours if we're doing a volume exclusion model, or they can interact with neighbours and have reactions if we're talking about a generic reaction diffusion type model. So that's the finest grain model, but it's most accurate, but it's also quite expensive, because if you've got n particles, at each time step you have to do n squared comparisons to find out where they are and who they can interact with. And you also need to make sure you take your time step, if you're doing a fixed time step algorithm, sufficiently small so that you don't miss interactions. Particles don't jump too far and pass the particles that they should be interacting with. So it can be very computation intensive to do these sort of microscale models. The next scale up, at the mesoscale, are models called compartment-based models. So you take the space that you're interested in and you discretize it into a number of voxels. It can be a regular lattice or it can be irregular, it doesn't really matter. And you allow the particles to jump between these voxels, usually just the nearest neighbour voxels. And then you allow them to react with other particles which are in their voxels. So this method is often called compartment-based or position jump or reaction diffusion master equation approach. And this is a sort of mesoscale model because you retain stochasticity, you retain some of the individual features of particles, but you lose the accurate information about where they are in space. And there are other problems with these in that they can be inaccurate because there are constraints on the sizes of the boxes you can take. You want the boxes to be sufficiently small that within each voxel the particles are well mixed, so you can just do usual mass action reaction kinetics on them. But if you take the boxes too small, then you disperse your particles too finely across the boxes and you lose bimolecular reactions. So particles just don't meet in the same box often enough to recapitulate the correct rates for bimolecular reactions. So there's limits on how big this box size can be, and sometimes you get the sweet spot, sometimes you don't in your model, depending on the parameters. So these models are much faster than the individual-based models, and they retain stochasticity, but they also have their own disadvantages. At the macro scale, we have things like PDEs, or stochastic PDEs, for example. So these can be much faster. If they're PDEs, you can sometimes do some analysis on them, so you don't even need to simulate them, which is great. But they don't do a great job of incorporating stochastic effects, and usually the models that you write down, PDE-based models, are phenomenological, so you've not really taken into account the fine-grained detail. The ideal is if you start with a fine-grained model and then derive the PDE from that, and that's how you can get a microscale model. Okay, so I've got some sort of cartoon pictures of what they look like. So this is my PDE, it's just a continuous curve. This is my on-lattice position jump model, the mesoscale model, so I divided space into these boxes, and I imagine the particles being stacked up on top of each other. Really, they're just well mixed in there, and they can all react with each other. And they can jump from one voxel to a neighbour, and the last one is this off-lattice position jump model, where I'm tracking the positions of the particles over time, and they can bump into each other or interact with each other. So that's the microscale or molecular-based model. I'm not going to talk so much about this one today, although we are working a little bit on interfacing these types of models. I just want to tell you a bit about two extensions to this on-lattice paradigm. So firstly, to speed things up, you can do non-local jumping, and I'll talk a bit more about this in a second, but you effectively if you're jumping back and forth between boxes doing diffusion, then it can be quite expensive because you're doing a lot of the same movements back and forth. If you allow your particles to jump further than just their neighbouring boxes, so to 2 or 3 boxes away, then things can get a lot faster. You don't need to do so many of these events because the particles are jumping further. So that's non-local jumping. And then for this on-lattice position jump process, an extreme case when the capacity of each of these boxes is just 1, it takes me back to my volume excluding process that I showed you in the very first part of the talk. So cells, only one cell can sit in each site. But there are ways of making that a bit faster. Maybe I double the size of each compartment and I also double the capacity. So I make it twice as big, so M is 2 here, and I can say that 2 particles can sit in each box rather than just 1. So this is called a partially excluding paradigm. Or I make it 4 times as big and I say 4 particles can sit in each one of these voxels. And again, I can speed things up because these guys are jumping much further than their fully volume excluding counterparts. Okay, so these are the sorts of models I'm going to be looking at. So the first hybrid model I'm going to tell you about is a local to non-local jumping model. So the first thing I need to do is establish this equivalence framework between local and non-local jumping. So to do that, for these on-lattice models, I can write down what's called a probability master equation, and then I can use that to derive the evolution of the mean numbers of particles in each box of the lattice. And so if I do this, then I find that for this diffusion paradigm on the local jumping model, with these particular rates, I find that my the equations which describe the evolution of the mean numbers of particles in each box just looks like a discretization of a PDE, and that PDE is the diffusion equation. Okay, so with these particular jumping rates, which are the standard ones for these on-lattice jump processes, I find that the mean numbers of particles in each box evolves according to a discretization of a PDE, which is the diffusion equation. So what I want to do is to make sure that if I allow Q to be greater than 1, so Q is the maximum length which I can jump in my non-local model, I want to do the same thing, write down the probability master equation, determine the mean equations for the evolutions of the mean numbers of particles in each box, and I want to choose these jumping rates so that I get back to a discretization of the diffusion equation in the same way. So it turns out that these are the choices you have to make in order to do that when Q is 2 and then when Q is 3. Okay, so in some senses I've established this equivalence framework that in the mean field, in the continuum limit, these models both describe diffusion, the diffusion PDE. The problem is that in my non-local jumping rates, you'll see that the total rate of jumping away from this box marked X is actually less. If you add these two jump rates up, it's actually less than the total rate of jumping away from this box marked X here in the local model. So if I have all my particles initially sitting in this one box, maybe it's an extreme initial condition, but it's some sort of modelling some sort of delta function. If I have all my particles sitting here, then the particles in the non-local jumping models will not jump away as fast as they should be doing. Okay? If things are smooth and spread out nicely, then you don't really notice this effect so much. And the reason for that is because although particles are jumping out of this box less frequently than they should be, they're also jumping in from these neighbouring boxes less frequently than they should be. So as long as things are quite smooth and well spread out, you don't really see these effects. But if you have steep gradients, then you really see a difference between the local jumping model and the non-local jumping model. So now I've got this equivalence framework. I also have a model which is expensive but accurate, and I have these non-local jumping models which are cheaper to simulate because I jump further, so I have to do fewer jumps, but they're also inaccurate in some senses. If I'm trying to model steep gradients, then these models fail. So this motivates a hybrid model. Okay, so I might want to put an interface, this thick black line, somewhere between regions where there are no steep interfaces on the left, so I can do non-local jumping, but maybe there's a steep interface on the right, and I want to get that correct, so I want to do local jumping in this case. So I'm not going to go into all the details of how you hybridize this, but very briefly, we did a bit of work on how to derive boundary conditions, appropriate flux boundary conditions for these sorts of non-local jumping models. So in fact, all you have to do is every time, this is the non-local jumping region. In the case Q is 4, so my particles can jump a maximum of 4 sites. So if they're jumping to the left, then they do their jumps with the usual rates that I showed on the previous slide. If they're trying to jump to the right, if they hit the interface, they just get reflected back. And instead of doing flux according to the non-local model, I just calculate how many particles there are in this box, how many there are in this box, and I know then what the flux should be over this interface because it's just diffusion. I can calculate what that flux is, and then I can implement one single special jumping rate for each of these boxes which is sufficiently close to the interface, and I can implement my flux boundary condition correctly across the interface. And similarly, if I'm jumping right, this is the local side now, and it's only this box which is affected. If I'm jumping right, then I use the usual jump rate. But if I'm jumping left, then I have to redistribute these particles into each of these 4 boxes, which is sufficiently close to the interface. So that's a sort of sketch of how this works. We make sure effectively that we're implementing the correct flux boundary condition over the interface, but more details can be found in this paper here. So it does work, and there are lots of simulation examples in there, but I don't really have time to show them in here. The second hybrid model I want to show you is from fully to partially excluding. So the fully excluding model, I've said you can have 1 cell per site at most, and in the partially excluding, I can have M. So in this case, M is 4. I have to somehow come up with a way of choosing the correct transition rates to make sure that this model, this partially excluding model, corresponds to this fully excluding model. Again, I won't go into too much detail about how to do that, but again, we use a probability master equation approach, and we try to make sure that the evolution of the density of these 4 aggregated boxes matches the evolution equation for the evolution of this single partially excluding box. The way we do that is to choose some sort of blocking probability. So if a cell tries to jump into this box, to mimic the volume excluding effect, I should be blocking some of these jumps from happening. They shouldn't all just be allowed to jump into here. So if the box is completely full, so capacity 4, if there are 4 cells in there, then the blocking probability should be 1. I shouldn't be allowed to jump into that box. If the box is completely empty, then I should just jump in with the normal rate. I should be allowed to jump in. There should be no hindrance. And actually, it turns out the correct thing to do is to use the blocking probability to be n over m, where n is the capacity of the box. Is the current occupancy of the box, and m is the capacity of the box. So it's basically just the fraction of free space which is available. That's how often I should allow these cells to jump into this box, which sort of intuitively makes sense. Okay, so then I can use these partially excluding models to significantly accelerate these simulations while still accounting for volume exclusion, because I can let the particles jump further. But imagine a situation where I've got particles of type A here and particles of type B here. Then in the fully volume excluding model, these guys will diffuse and eventually they'll bump into each other, but they won't be allowed to cross over each other, right? In the fully excluding model, they shouldn't be allowed to jump over each other, because that would mean that one box would have capacity greater than 1. And so every jump attempt where one tries to jump into the other should be avoided. So you'll never find a B over here and an A over here. But in the partially excluding model, because these boxes have capacity greater than 1, these cells spread out, and then you can have an A and a B in the same box, and they can swap over. So in some senses, this fully excluding model, although quicker, is not actually accurately modeling what's happening if I'm talking about a single file diffusion and exclusion process. So what I need to do is have regions over here where A and B can jump quickly using the partially excluding process, but where they might come and meet each other, I need to use the fully volume excluding process so that I don't have A and B particles crossing over each other. So that again motivates a hybrid model, and the way we do it in this case is using something called a hybrid box. So over here we're doing fully volume excluding, we've got this fine grid. Over here we're doing partially volume excluding, we've got this coarse grid. But in here we're doing a bit of both. So particles can jump according to the fine, fully volume excluding process in here, and a particle gets into this box from the right by just jumping in according to the fully volume excluding process. But particles can also jump into here. So any particle in here can jump into the into the partially excluding box with rates which are taken from the partially excluding case. And similarly, you can have partially excluding particles, or particles in the partially excluding box jumping into here, and again the blocking probability is chosen according to the occupancy of these sites. So if a cell is allowed to jump into here, then we just place that cell uniformly in one of the empty sites in this hybrid box. So this method turns out to work, and you can prove that it works using the same sort of mechanism that I discussed before, using a probability master equation. Okay, so again, I'm not going to show examples, but there's a couple of papers where you can read a bit more about this. The last 2 methods I want to talk about are going from a PDE to a compartment-based method. So this first one is something I call flux balancing. So in this region of the domain, I'm doing purely compartment-based simulation. In this region, I'm doing purely PDE, but I have these two interfaces here. And in the region between these two interfaces, mass is modeled both as PDE and an equivalent description of the particles is using this compartment-based method. And so what I need is at this left-hand interface, I need a boundary condition for the compartments, and this right-hand interface, I need a boundary condition for the PDE to make sure that my model is well specified. So the boundary condition for the PDE is I just implement some Dirichlet boundary condition where I say that I fix the density of the PDE at this point to be the average of the number of particles in this compartment and the number of particles in this compartment. So it just sits somewhere exactly in between the heights of these two boxes. At the left-hand side, I need to have a condition on the compartments, and so all I do is I calculate the gradient of the PDE, and that tells me the influx of particles that should be occurring into these compartments. So I implement that influx by adding particles in stochastically to make sure that on average I'm matching that flux. And so by coupling both the actual value of the PDE and the flux of the PDE to the actual value of the compartment and the flux in the compartment, I ensure that these two models stay in sync. Basically, they maintain the right profile. And again, I don't have time to show examples for this one, but you can read a bit more about it in this paper in Journal of the Royal Society Interface, which came out a couple of months ago. So the very last one I want to tell you about is one which is quite a generic mechanism for coupling these different sorts of algorithms, and it's called a pseudo-compartment method. So again, on the left, I have PDE, on the right, I have compartments. This time I just have a single interface at 0. And the idea is that in the region of the coarse-grained model, in this case the PDE, I have a region which is called a pseudo-compartment, which is both PDE and compartment. So I use a zero flux boundary condition on the PDE here to make sure no PDE mass can flow over the interface. I'm not losing mass according to that. And then I make sure that the only way I can do jumping over the interface is according to the compartment-based method, so the stochastic method, the finest grain method in this case. So I can have particles jumping out of this compartment and jumping into here, and if that happens, then all I do is I increase the number of pseudo-particles that I have, and I add a boxcar function worth of mass, a single particle's worth of mass as a step function to the PDE. Similarly, I calculate the number of pseudo-particles I have in this box by just integrating over the PDE, and then I have a jumping rate out of this compartment into the other one. And if a particle jumps out of this compartment into the first compartment of the compartment-based method, then I just remove a particle's worth of mass as a step function from the PDE. I should say this algorithm, this is the simplest explanation I can give of this algorithm. We have a much more complicated version of the algorithm which works even when you have very low populations of particles in the PDE regime. The problem with removing a step function from this PDE is that you have to have enough PDE mass to do that. You don't want to send the PDE negative. So if you've got low population numbers at the interface here, then this algorithm can fail because you're trying to remove a step function's worth of mass from the PDE, but you just can't do it. So we have a method which allows you to correct for that. But arguably, I would say you shouldn't be using the PDE in the region where you have low particle numbers anyway. But that's a sort of philosophical argument. So I'll show you to finish a few of these simulations to show you that this does actually work, even though I haven't managed to show you any of the other methods that work, but I promise you they do. So this one, I'm going to try and maintain a uniform particle profile. So this is the first thing I get my students to do whenever I give them a challenge to build a hybrid model is, can you maintain a uniform particle profile? And the answer is usually no. Usually you see this big disparity between the PDE side and the compartment-based side. So what you're seeing here is red is the analytical solution of the mean field PDE. So in this case it's just constant because it's diffusion. This is the steady state. And the blue is the compartment-based part of the hybrid method, and you'll see a blue curve on the left-hand side of the domain, which is the PDE part of the hybrid-based method, but it will hopefully sit around the red curve for most of the time. So you can see there is stochastic variation. The PDE does vary because it's effectively subject to a stochastic boundary condition, but overall the mean behavior of this hybrid method should be matching the mean behavior of the mean field model, which is the red curve. So that's just a by eye comparison, but we can do some more slightly more rigorous stuff than that. So we can just compare the amount of mass in the PDE region to the amount of mass in the compartment-based region, and in the mean field it should just be half and half. The mass should be zero flux and zero net flux over the boundary. And you can see, so the black curve is the hybrid model, and you can see firstly these values are quite small. The more repeats I do, the smaller these values get. And although it looks maybe like there's slightly more mass in the PDE region here at the end than there is in the compartment-based region, or vice versa in fact, if you run these for long enough, you see that this black curve just oscillates around the red curve. There's no systematic bias in the mass and where it is. So it's good news. It looks like we can replicate a uniform gradient quite nicely. Why you would want to do that, I don't know, because it's completely ridiculous, but it's a useful test case. The next thing we want to do is to check that we can model flux correctly. So we wanted to do the biggest flux we could possibly do, and with diffusion, since the functional form of your flux is stipulated already, the only thing you can do is to put all the particles in one regime and none of the particles or none of the mass in the other regime. So put all the mass in the PDE regime, and we want to see that again we can make sure we have the correct profiles as time goes on. So I'll just play the movie, and you can see again that at least by eye the hybrid method seems to be matching the analytical solution of the mean field PDE, the mean field diffusion equation. Okay, so here, this is the actual solution of the Fourier series solution of the diffusion equation with that initial condition. And so the red curve is the amount of mass you'd expect to see, or the proportion of mass you'd expect to see in the PDE region, and the black curve is the proportion you actually do see in the hybrid method. And again, you can plot things like relative error, which is where you subtract the red curve from the black curve and divide by the red curve, so you can get some sort of relative error. And you can see again, this has no long-term systematic bias in where the mass sits in this simulation. So we say that the hybrid model is doing a decent job of replicating the amount of mass in each region, of doing diffusion properly. The last one which I want to show you is one where we come up with an error in the first algorithm that I told you about, the most simple version. It's a morphogen gradient formation example, so taken from sort of developmental paradigm. We have the diffusion equation, we have decay of particles, and then we have a flux boundary condition at the left-hand side. So it says a certain amount of mass is flowing in. And we have a zero flux boundary condition on the right-hand side, and an initial condition with no mass in the system. I'm going to just run this video. I'm going to pause it quite early on, hopefully. So here you can see that the hybrid method is systematically overestimating the amount of mass in the PDE regime, especially near the interface. And this is exactly that problem I described to you. When you have low amount of PDE mass near the interface, you can't remove a step function's worth of mass, and therefore you can't create a particle. So the flux over the interface is retarded. So you can't do the right flux over the interface. You see this error. It looks, as you run the video, it looks like it corrects itself, but actually it's actually always there for the duration. So that's bad news for the simple algorithm. You can see it's not doing a terrible job, but there is an error, and you can see it most clearly here when I do the same sorts of error plots. You can see that the hybrid method underestimates the amount of mass that you would expect to see according to the mean field model. But the good news is that with a revised version of the algorithm, where you rescale the PDE to take a particle's worth of mass out rather than just taking it from the first box, then you can get these things to match up quite nicely. And again, you can see that the relative error has no systematic bias as time goes on. Okay, so that's just the simulations to prove that at least one of the things that I've told you today was vaguely true, even if the other ones weren't, which they are, but you can read the papers if you want to know a bit more or talk to me afterwards. So I'll just finish by talking about where we're going to go next with this sort of stuff. I've got a student currently who's just been doing his master's with me. He's just started a PhD with me. He's working on growing domains. So I initially motivated this part of the talk by saying, well, looking at mouse embryos, and they grow in time. So we need to be able to do these sorts of hybrid method on growing domains, and it's non-trivial to do that sort of extension. So he's just writing that up at the moment. There are other coupling methodologies. I've told you about a few that we've come up with, but there are lots of other coupling methodologies which we would like to investigate and see whether they work better or worse. In particular, we're interested in not just the mean field behaviour of these models, but making sure that we don't affect the variance of the stochastic model, because often that's an incredibly important aspect of the stochastic model. That's why you have a stochastic model in the first place, because it can incorporate noise, and you don't want to change the properties of the stochastic noise. So we need to make sure that our hybrid methods aren't doing that. We have an idea of how to do PDE to off-lattice based hybrid methods using similar sorts of paradigms. And also, I haven't shown any volume exclusion to PDE type simulations, which would really help to speed up those volume exclusion type simulations. Fully to partially, or partially to fully excluding volume excluding hybrid models can significantly accelerate things, but PDE to volume excluding could do even better. And like I've sort of said, hinted at throughout, obviously the last reaction, the morphing gradient, was reaction diffusion, but it's never the sort of reaction diffusion which will give you patterning or Turing patterning, if you like. So you need to have higher order interactions, bimolecular reactions, if you want to do that sort of thing. And at the moment, we don't have a robust way of incorporating higher order reactions into these sorts of hybrid systems. So that's something we'd like to incorporate as time goes on. So this is just a summary of what I've talked about today. It's in two parts. There was the first part, which is the biological background. We showed that actually a relatively straightforward mathematical model can give you some really interesting biological insights. And the second part was motivated by that, talking about how to develop these hybrid methods, different modelling paradigms, and how to come up with algorithms for how you can hybridize these two different types of modelling regime. And I tried to show that at least in some cases they actually work. So I'll finish by saying thanks to the people who've worked on this project with me. I've got students in Oxford. Paul Taylor's just handed in his thesis, so he'll be finished soon. Jonathan is a second-year PhD student who worked with me on a summer internship project, and Cameron is just starting with me in Bath at the moment. And my collaborators are Ruth Baker in Oxford, Matt Simpson at QUT, and Mark Flegg in Monash. And thanks very much for coming along and listening to me waffle. Thanks. Cheers.
Thanks, Kit. I think we have 3 minutes before people might have to run off to meetings. So has anyone got any questions for Kit?
When you talk about growing domains, is that modelling the fact that the animal is growing, the embryo is growing while the diffusion is occurring?
Yeah, exactly, yeah.
So what resolution is the domain to start with? I mean, you talked in the second part about the performance aspect of things, but it wasn't clear to me how slow the thing is in the first place and how many millions of cells there are, voxels.
Yeah, that's a good question. So I don't actually know how. I guess there were order of thousands of, tens of thousands of voxels, and therefore by the time the cell gets fully populated, we're talking tens of thousands of cells. But I guess it depends what animal you're modelling, but I guess I could figure it out from the final domain length readouts and the size of the voxels.
And modelling a human is harder than a mouse just because you end up needing a bigger resolution.
Yeah, the mechanisms are broadly similar for the movement, although obviously less is known because it's slightly less ethical to do experiments on humans than a mouse. But we assume the mechanisms are broadly similar in all mammalian embryos. But yeah, human embryos. So my Vitruvian man to Vitruvian mouse was slightly misleading because at the embryonic stage, the embryos are not that at the stage that we're interested in. The embryos are not that different in size, but the human embryo is obviously still bigger than the mouse embryo. And actually the time period over which these cells colonize is also quite different.
My third question.
Yeah.
The implementation was what you were implementing cellular automata in MATLAB.
Yeah, that was MATLAB, but for the actual experimental paper, we did it in C++. So yeah, we've already exhausted my computational efficiencies by going to a high-level programming language. But yeah. But yeah, MATLAB is very useful for prototyping. So the simulations I showed at the end are just done in MATLAB. But yeah, for the actual on-lattice simulations of the mouse, we were doing that in C++.
Any other questions? I have one, if not. I was kind of curious about what are the implications now that we understand better that piebaldism is caused by defects in proliferation instead of migration? What can that tell us about the kind of more serious biological conditions you were referring to?
Yeah, it's interesting. I think each of those cases has to be taken on its merit. So I don't think that we could say because melanocytes or this KIT mutation changes the way that cells migrate in a particular way that that's necessarily what happens in the defects in migration of neuroblasts. But I think the idea is that we can use some of the modelling techniques and tools that we've developed to understand how melanocytes and melanoblasts are moving around, to try and understand how neuroblasts are moving around in the same way. So the conclusion itself is not directly applicable to a whole range of different diseases, but the modelling paradigms that we've developed to try and understand those systems are. Yeah.
And so insofar as I understand lattice modelling and off-lattice modelling, the kind of lattice model you developed is quite sophisticated. And what are you expecting to be the benefits of going to an off-lattice scheme instead?
Yeah, so there are certain things that happen in on-lattice models which are just not really realistic. So cells, if they're next to each other in an on-lattice model, they just can't jump past each other. That's the volume exclusion paradigm. But actually, if they're in an off-lattice model, they can somehow go around each other more. They've got more degrees of freedom to move around each other, and that is what happens with real cells. They don't just sit there and block each other as they do in this on-lattice model. They can actually work around. They can deform and move around each
So
other.
would a halfway point be to move to 3 dimensions in your on-lattice model first before jumping to off-lattice?
Yeah, so that's something that we're thinking of doing, especially because these cells actually, after a couple of days of migrating around in the dermis, they then move up to the epidermis, which is a different layer of the developing skin, if you like. So yeah, although our three-dimensional sort of, the cells don't tend to sort of crawl over each other, if you like, or there's no evidence of that, but they can move in 3 dimensions, so they can get around each other in more ways than just they could do in 2D. So yeah, certainly 3D to model these 2 different layers of the skin is going to be quite an important extension to really understand what's going on.
Okay. Any last questions? Oh, right, hold on, it's past 3, so I feel bad. I should let everyone go on to other meetings. But for now, can we just say thank you again to Kit for his interesting talk?
Thanks.